Good morning, students. So we are going to continue with the chapter 6 of Science 2 of 9th standard, that is heredity and variation. Okay. So in the previous lecture, we have studied about heredity, inheritance, genetics, inherited traits and expression of traits, chromosomes, and types of chromosomes. In today's lecture, we are going to study in details about DNA. So this is the structure of the DNA. All of you can see on the screen. This is how the DNA looks like. What is actually DNA? DNA is deoxyribonucleic acid, right? It is also called as the master molecule. This is the molecule which is present inside the nucleus of our cell. In each and every cell of our body, this DNA is present. So it is not only present in human beings, this DNA is present in all the living organisms. If we say that the organism is living, that means the DNA is present in that organism, right? So it is present in bacteria, viruses, plants, animals, human beings, in all the living organisms. So let us first see how this DNA was discovered. So this DNA was discovered by the scientist, who was called as Friedrich Mischer, right? So you can see here. So this Friedrich Mischer, he began working with white blood cells in 1869. So he was working on the white blood cells. You know that in blood there are different types of cells. That is RBCs, that is red blood corpuscles, white blood corpuscles, right? So in he was studying about WBCs, that is called as white blood cells. So white blood cells are major component of pus and infections, right? So when some infection is there and the pus is there in that infection, so white blood cells are present in large amount in that pus. So as a result, Misha collected a lot of pus from bandages at a local hospital. So and then what he did is he added a weak alkaline solution to the white blood cells. When he did, the cells decomposed and caused the nuclei to move out of the solution, right? So as soon as he added the weak alkaline solution to those pus cells, so the pus cells, they decomposed and the nucleus come out of the solution. So come out in the solution. From the nuclei, Misha isolated a substance known as nuclein. So from the nucleus, he found some substance coming out that he called it as nuclein. After chemical analysis, nuclein was later renamed as DNA. So after when the chemical testing takes place, then they found that DNA is deoxyribonucleic acid. That means the components of the DNA or the components of that nucleic acid material was deoxyribonucleic acid. So that is why they named it as DNA, right? So this was the first person to identify DNA from the nucleus, Friedrich Mischer. So as I've told you, the DNA is present in bacteria, viruses, plants, animals, human beings, in all the cells. So let us see the location of the DNA. So you can see cell is here, right? This is an animal cell. So inside the cell, there is nucleus. Then inside that nucleus, there are chromosomes. And when the chromosomes are a compact coiled structure. So when you open that, uncoil that chromosomes, you will find the DNA. So DNA are present in a highly coiled form inside the chromosome. It forms a structure that is called as chromosome. So chromosome is nothing but it is formed of DNA and proteins, right? And it is highly coiled and it is present inside the nucleus, which is present inside the cell. So this is the location of the DNA. Cell, inside cell it is nucleus, and there is chromosomes, and these chromosomes are made up of DNA. Okay, so everybody should be knowing where this DNA is present. So here we'll try to understand the uncoiling of the chromosome, right? So here you can see, as I've told you, the chromosomes are present inside the nucleus. So this is the structure of the chromosome. This is chromosome. And when you magnify that chromosome, you can see such kind of structure. As you must have seen previously, there used to be a telephone wire. The landline telephone used to have such kind of wire. So in this way, it is coiled, and inside that the DNA, the thread-like structure is present, that is called as DNA. So this DNA is coiled around the proteins called as histone proteins. This ball-like structure which you can see, these are histone proteins, right? So some histone proteins is gathering like this, and around that this DNA is coiling, right? And it is highly coiled. And see, you can see here, the DNA is coiled around these histone proteins. Then it is also coiled around these histone proteins, and that way it is highly coiled, and it is then placed inside the chromosomes, right? So chromosomes are mainly made up of DNA and the histone proteins. So here you can see. Now let us study about DNA. What is the full form of DNA? DNA is deoxyribonucleic acid. Okay, DNA is deoxyribonucleic acid. Chromosomes are mainly made up of DNA and proteins. It was discovered by Swiss biochemist Friedrich Mescher in 1869 while he was studying white blood cells. Then DNA is present in nucleus, and that is why it is called as nucleic acid. It is present in all the organisms from viruses and bacteria to human beings. As it controls the functioning, growth, and division of the cell, it is called as the master molecule, right? So this DNA, it controls the functioning of the cell, all the functions of the cell, whatever cell is carrying out the function, all the functioning of the cell is under the control of the DNA. The growth of the cell is under the control of the DNA, and even the cell division is under the control of the DNA. So that means the whole control of the cell is done by DNA. So that is why it is also called as the master molecule, right? So it controls functioning, growth, and division of the cell, and are therefore called as the master molecules. Right? So you should be able to say about DNA. Now you know what is DNA, right? So without this also, now it should be filled in your minds that what is DNA? The full form of DNA is deoxyribonucleic acid. It is present inside the nucleus of the cell. It is present in all living organisms, and it was discovered by Frederick Mesher. And it also it controls the functioning, growth, and division of the cell, and therefore these are called as the master molecules. Right? Then let us study more details about DNA. Yes, the structure of the DNA. Now the next point is structure of the DNA. In 1953, Watson and Crick, you can see the pictures. These are the scientists, Watson and Crick. They produced the double helical model of the DNA molecule. So these were the people, they have found out how the structure of the DNA is, right? So they have studied the structure of the DNA, and that is why the structure is now called as Watson and Crick double helical structure, right? It was discovered in 1953. So the structure of the DNA molecule is the same in all organisms. So in 1953, Watson and Crick produced a model of the DNA molecule. As per this model, two parallel strands of the nucleotides are coiled around each other. So let us see the structure. Have you seen the photos of Watson and Crick? And in front of this, you can also see the double helical structure, which is placed in the model of the DNA. Here, this is the model of the DNA, and these are the scientists Watson and Crick. Okay. Now let us see. Let us study in details the double helical structure of the DNA, what they have found in that. As per this model, two parallel threads of nucleotides are coiled around each other. So you can see here, see, this is one strand of DNA. This is the other strand of DNA. So they'll run parallel to each other. So as per this model, two parallel threads of nucleotides are coiled around each other. This arrangement is called as the double helix. This structure can be compared with a coiled and flexible ladder. Right? You must have seen a ladder. When you coil that ladder, you will get such kind of structure. Each strand in the DNA, in the molecule of DNA, is made up of many small molecules known as nucleotides. Right? So here we will see the components of the DNA molecule. See here, this backbone or this helical structure, what you can see, this backbone is made up of sugar and phosphate. What are the chemical components of this backbone? Sugar. It is made up of sugar and phosphate. That is why it is also called as sugar phosphate backbone, right? And inside this, the steps which you can see, as we have steps in the ladder, the step-like structure which you can see, those are called as nitrogenous bases, right? So there are four kinds of nitrogenous bases in the DNA, that is adenine, thymine, cytosine, and guanine. Right? So there are four types of nitrogenous bases: adenine, guanine, cytosine, and thymine. Adenine and guanine are called purines, while cytosine and thymine are also called as pyrimidines. So in the structure of a nucleotide, a molecule of nitrogenous base and phosphoric acid are each joined to the molecule of sugar, right? So sugar is joined with phosphate and the nitrogenous base. As there are four types of nitrogenous bases, nucleotides are also of four types. Nucleotides are arranged like a chain in a molecule of DNA. So let us see. Here we have seen the structure. So there are four types of nitrogenous bases. Adenine, guanine, right? So the nitrogenous bases, you should know the names. There are 4 types of nitrogenous bases: adenine, guanine, cytosine, and thymine. Adenine and guanine are called as purines, while the thymine are called thymine, and cytosine are called as pyrimidines. So in the structure of the nucleotide, a molecule of nitrogenous base. So you should know the component of the DNA. It is made up of nitrogenous base, that is ATGC, adenine, thymine, guanine, cytosine. Those are called as nitrogenous base. It is also made up of phosphoric acid and the sugar molecule, right? So sugar, phosphoric acid, nitrogenous base are the components of the DNA. So as there are 4 types of nitrogenous bases, nucleotides are also of 4 types. And always you should remember, when you're showing the combination of the nitrogenous bases, adenine always pairs with thymine and cytosine always pairs with guanine. That is ATGC. So always adenine combines with thymine and guanine will combine with cytosine. ATGC pairing is there. And when these are called as the nucleotides, when these nucleotides are paired, for example, now 18. Now let us see another example. If we are having the combination of ATGC, right? So when 3 nucleotides are this way, that is called as a gene, right? So the combination of the 3 nucleotides forms a gene, and one gene is responsible for the synthesis of one type of protein. For example, if we have to secrete a growth hormone, right? So this growth hormone is nothing but a protein. So it is going to be synthesized with the permission of the gene, and this gene is present on the DNA, and it is present in the coded form. And these 3 nucleotides, they are arranged in such a way that this 3 nucleotide combination is a gene, and it is responsible for the synthesis of one kind of protein. Now let us see. So this was just an example, ATGC. So this may be responsible for the growth hormone, right? So it is not that way, but this is just an example that the 3 nucleotide combinations are responsible for the protein synthesis. So it is also present in different way. It can be ATGC, AT, right? So this also forms a gene. It can also be GC, 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 3 GCs together, right? So that way many different combinations are possible. So that means many different genes are present, and these are in the coded form, which gives the information for the cell to work. Then here you can see the detailed structure or the molecular formula of the DNA. See, this is a sugar molecule. You know the molecular formula of a sugar, right? So this is the structure of this sugar. Then this structure is, this sugar molecule is attached with the phosphate group. This is called as the phosphate group. And also the sugar is attached with the nitrogenous base. This is the structure of the adenine, right? So sugar attached with phosphate group and sugar attached with nitrogenous base. All these 3 together forms one nucleotide. This is only one nucleotide, right? Sugar phosphate backbone is there, and the nitrogenous base is also attached to it. So I hope that the molecular structure is followed by you. You understand it. Okay. Now here you can see more details. You can see here molecular DNA, double stranded DNA. So see here, this is the sugar phosphate backbone. This is sugar molecule. This is phosphate. This is sugar molecule. This is phosphate group. So this is the sugar phosphate backbone. Here on the other side is also sugar phosphate backbone. This is called the 5' end, and this is called as the 3' end. So this 3', 5' you will understand later in the bigger classes because it is attached on that 5th carbon atom, and the down it is attached on the 3rd carbon atom. So that is why it is called as 5' and 3' end. And see here, this is the nitrogenous base, right? So this nitrogenous bases, see, this is AT, this is GC. So these nitrogenous bases are joined with the help of hydrogen bond, right? So this is how the double helical structure of the DNA molecule appears, or it is formed. Right? Sugar phosphate backbone, nitrogenous base, and they are bonded together by hydrogen bond. Then, as I've told you, gene. What is gene? So genes are nothing but the segments of the DNA molecule. Segments of the DNA molecule are called genes. Due to variety in the sequence of the nucleotides, different kinds of genes are formed. These genes are arranged in a line. Genes control the structure and the function of the cells and of the body, right? So when it controls the structure and the function of the cell, indirectly it also controls, when it controls the structure and function of each and every cell, that means indirectly it controls the structure and functioning of the whole body. Also, they transmit the hereditary characteristic from parents to offspring, right? So at the time of meiosis, the crossing over takes place and the genes are being crossed over, and the genes are being transmitted from the parents, right? Hence, they are also the functional unit of heredity. Okay. So we'll highlight the major points. The segments of the DNA are called as genes. And due to variety in the sequence of the nucleotide, as I've told you, there are different sequences of the nucleotide. It can be AT-AT-GC, it can be AT-GC-GC, it can be GC-GC-AT, it can be GC-AT-AT, or you can make thousands of combinations with this 4 nucleotides, right? If you want to try, you can try on your own. And 3 nucleotides forms 1 gene, and which is the coding for the protein synthesis of our body. Then these genes are arranged in line. Genes control the structure and the function of the cell, and that is why they are the functional unit. Also, they transmit the hereditary characteristics, right? Okay, then next point is Human Genome Project. So now the scientists have, as the DNA was discovered, that something is there which controls the cell. So that is called as the DNA. Then DNA was discovered. Now the scientists wanted to know the sequencing of the DNA. That means what genes are present at which place, right? And which gene is responsible for which type of protein. So that is what is called as the Human Genome Project. That is finding out the sequence of gene on the DNA. So in 1990, the Human Genome Project was together undertaken by all the geneticists of the world. In June 2000, scientists of this project and Celera Genomics Corporation, there is a Celera Genomic Corporation, a private industry in USA, collectively announced the discovery of the complete DNA sequence of the human genome. Depending upon the findings of this project, scientists confirm that the number of genes in the human genome is about 20,000 to 30,000. Right? How many genes are present on our DNA? Almost 20,000 to 30,000 type of combinations. I've told you the nucleotide combination. So almost 20,000 to 30,000 genes are present on our DNA. And the scientists have discovered that. Later, scientists discovered the genomic sequence of many microorganisms. Also, they have done the sequencing of many microorganisms. So what is the use of this Human Genome Project? What is the use of knowing the sequencing of the DNA? Can you think of? Yes. Due to the research in genomics, disease-causing genes can be identified. If disease-causing genes are identified, genetic diseases can be diagnosed and it can be properly treated. Isn't it? For example, the diabetes, it is a genetic disorder, isn't it? Some kind of problem is there with the genes. So now if we know that what kind of, where is this gene located, which is mutated or which is not functioning properly, and it is causing diabetes in the body, right? So if we know the sequence of that gene, if we replace those gene, and then we can treat the diabetes, right? In this way, the other diseases are also there. There are many different types of genetic disorders, right? For example, albinism. If we know that there's some gene which is responsible for producing the melanin pigment of our body that is not functioning properly, right? So that gene can be replaced, or some treatment can be done on, and that disease can be treated. So in this way, the different types of genetic diseases which are there, for example, you must be heard of the Down syndrome, right? Or the Turner syndrome. So these are the genetic diseases. So if we know that where is the problem in the gene, and if we can treat that problem, then it would be good for us. We can treat the genetic diseases also, as till now it was there that the genetic diseases were difficult means almost impossible to treat. If it is given, genetic disease means the disease which is given by nature, right? But if we will do the research, if we'll continue the research on this, so at least we can treat some percentage of such kind of diseases. Okay. Also, there is DNA fingerprinting. There is a topic called as DNA fingerprinting. So the sequence of the genes in the DNA of a person, that is the genome of a person, is identified. Now the sequencing of the gene on the DNA is unique. Unique means it is different for each and every individual. No two individual will have the same kind of DNA. It's not possible. It is impossible, right? So the sequencing on the DNA is different. So the DNA of each and every individual, of each and every animal, each of every human being, each individual is having a different DNA, right? So the sequencing of the gene in the DNA of a person, that is the genome is identified, and what it is used nowadays, that is called as DNA fingerprinting. So it is useful to identify the lineage. Lineage means who was your father, your grandfather, your grandfather. So that kind of, you can identify the lineage. And also it is used to identify criminals because it is unique to every person, right? So nowadays this DNA fingerprinting is used in the investigation purposes in CID, in CBI, to identify the criminals, right? You must have seen in Crime Patrol or the other serials how they send it for DNA fingerprinting, or they say that we need to do this DNA testing, isn't it? So here we finish with the topic. So let us revise quickly what we have studied today. Okay, so yes, so today we have studied in details about the DNA. So DNA was first identified by Friedrich Mescher in 1869 from the white blood cells, right? So then it is present in this DNA is deoxyribonucleic acid. It is present inside the nucleus. It is present in all living organisms, bacteria, viruses, plants, animals, human beings. Then location of the DNA, it is present in each and every cell inside the nucleus in the form of chromosome. It is present in highly coiled form, right? It is coiled around the histone proteins, and it is highly coiled, and it is filled in the compact form in the form of chromosome, right? So in short, quickly, let us see. DNA is deoxyribonucleic acid. Chromosomes are made up of DNA and proteins. It was discovered by Friedrich Mescher. DNA is present in the nucleus. That is why it is called as nucleic acid, present in all viruses, bacteria, plants, animals, human beings. And it is called as the master molecule as it controls functioning of the cell, growth of the cell, and division of the cell. Then structure of DNA. So in 1953, Watson and Crick produced a double helical model of the DNA molecule. It is called as the double helix. So here you can see, as per this model, there are two parallel strands. It is made up of sugar and phosphate backbone. And between those sugar phosphate backbone, there are base pairs present, nitrogenous bases. It is also called as the double helix structure. And each strand of the molecule of DNA is made up of many small molecules known as nucleotide, right? So the DNA is made up of nucleotides. One nucleotide is made up of sugar, phosphate, and nitrogenous base. So there are four types of nitrogenous bases present in DNA: adenine, guanine, cytosine, and thymine. So adenine always combines with thymine, and cytosine always combines with guanine, ATGC. Right? Those are also called as purines and pyrimidines combination. So there are four types of nitrogenous bases also. This is the molecule of one nucleotide of DNA. You can see the structure of nitrogenous base, structure of sugar, and the structure of phosphate group. This is the molecular picture of the DNA, molecular DNA, right? Sugar, phosphate, and nitrogenous base, and they are bonded together by hydrogen bond. So that was all about the structure of the DNA. Then the next part is gene. So the small segment of the DNA is called as the gene. Due to variety in the sequence of the nucleotide, different kinds of genes are formed. These genes are arranged in line on the DNA. Genes control the structure and the function of the cell of the body. They also transmit the hereditary characters from parents to offspring. So they are also called as the functional unit of heredity. So we have also seen the Human Genome Project. So the scientists have identified that 20,000 to 30,000 genes are present on the human genome. And also we have studied about DNA fingerprinting, how it is used to identify the lineage, and also it is used to identify the criminals, as the DNA of each and every person is unique. So here we finish with our lecture. If you're having any doubt, you can ask.